Hello everyone, I hope everyone is well. My name is Fergus Preston and I'm currently a second year PhD student based within the Drug Discovery Unit at the School of Life Sciences at the University of Dundee. My project is on the identification of novel heterocyclic fragments for use in fragment-based drug discovery programs. So I'd first just like to thank the organisers for putting on this webinar conference. I think it's an invaluable opportunity both for myself to promote the research that I'm doing at the moment, as well as for you as potential students to understand the sort of chemistry, the wide range of chemistry that we do within the School of Life Sciences, and the benefit of carrying out a PhD at one of the top life sciences institutions in the UK. So to put my project into some perspective and background, so many of the drugs currently on the market have evolved through this high throughput screening methodology, where you take these relatively large, bulky molecules with quite a high molecular weight of 500, and you screen them against a protein of interest. From there, you'll then probably get an affinity, that being it's forming a binding interaction with the protein, and your affinity will probably be within the micromolar to nanomolar range, so very potent for a drug-like molecule. What a chemist would then do would be to take this hit and break this molecule down to really understand which part of this molecule is giving you the potency. This can be quite time consuming. So for example, it could just be this functionality here where that is giving you the potency and nothing else is needed. And so you'd then just focus on this functionality and build the molecule up again to hopefully improve physiochemical properties, that being the binding and the potency, whilst mitigating any toxicity issues. And this can take months, usually years, to fully optimize a hit to a potential drug-like lead. Fragment-based drug discovery has evolved where you take these smaller compounds, denoted as fragments, with a molecular weight of less than 300 Daltons, and you screen them against a protein of interest, so again similar way to high throughput screening. However, this way the fragment will bind and show an affinity towards the protein, but unlike high throughput screening where the chemist will then have to break this molecule down to understand which part of this molecule is giving you the potency, you as a chemist will be able to understand and know that if you get potency, it is directly correlated to the fragment that you're seeing. Thereby, you can do a step-by-step optimization to increase the size and potency of this molecule to obtain a highly potent, high affinity drug-like molecule, whilst mitigating any negative and desirable characteristics. So if you almost look at this Lego-like structure down the bottom here, again you take these fragments, you screen them against the protein, some may bind. You can then do further optimization and optimize this fragment into a good lead-like structure. The benefit of fragment-based drug discovery over high throughput screening is its coverage of chemical space. This is noted quite nicely where take 1,000 fragments with a molecular weight of 190 Daltons can actually cover the equivalent chemical space of 10 to the 8 molecules with a molecular weight of 280. This goes up to equivalent chemical space of 10 to the 18 molecules with a molecular weight of 440. So when you kind of get into 440 and 280 Daltons, you're really falling within high throughput screening, and therefore the issues that I mentioned earlier on associated with high throughput screening. There have been successes of fragment-based drug discovery. So there are 2 drugs currently on the market and a plethora of other drugs currently within different phases of the drug discovery pipeline. But as you can note here, whilst these drugs look relatively big and complex, highlighted is the initial fragment that showed the potency, and in all cases within fragment-based drug discovery, that initial fragment is somewhere within the final drug-like molecule. Therefore, as I mentioned on a previous slide, you can do a step-by-step optimization to optimize this fragment into a drug-like molecule that you see here. Whilst fragment-based drug discovery and indeed high throughput screening is good, many of the compounds fall within the similar sort of chemical space, and therefore, both as a chemist and a scientist, we're not actually exploring much chemical space as we would like. You can see here there's quite a lot of underexplored 3D structural diversity. So kind of as scientists, we need to ask ourselves, Are we missing out on potential new drug classes and treatments because we're not exploring this chemical space? And then predominantly for my PhD, how best can we explore this chemical space? So I identified a range of compounds. So I took 26 million compounds and filtered based on desirable characteristics that I would look for within the fragment. So discussions with my supervisor and my industry sponsors, so the sort of fragments and functionalities that we would like. And this yielded us with 11,500 undocumented and unreported fragments anywhere in the literature. Further filtering was then applied such that we had good synthetic handles, that being handles that we can do further elaboration on once we've synthesized the compounds and screened them against the library. We can then optimize it up into a much larger drug-like molecule. So I've synthesized 4 molecules at the moment, and I'm currently identified and synthesizing other molecules. And once I've got a good library size put together, I will combine these and screen against a target of interest or a protein of interest, hopefully get some affinity, and then do further subsequent chemistry to build that fragment up into a much larger lead-like compound. As for interactions within the school and industry, the School of Life Sciences is in quite a unique opportunity, and I don't know many places where, as a scientist, you can share your ideas with other members of the school via a weekly seminar series. And as for myself, based within the drug discovery unit, I'm gaining invaluable experience of the drug discovery process at the early stage of my career, from which I can then expand my scientific knowledge both experimentally and theoretically. And the drug discovery unit, and indeed the School of Life Sciences, is a very multidisciplinary environment whereby biologists, chemists, computational chemists, and drug metabolism scientists all working together, either in the same office and lab, or definitely in the same corridor. So this is a benefit, such as myself as a chemist, I can synthesize these compounds, give it to our biologists to screen. Once they've come back with the results, I can then optimize my fragment and build it up systematically into a much larger compound. And then I can give it to a drug metabolism scientist, whereby they can do all of the animal testing. So we have a fantastic animal testing facility based within the School of Life Sciences, where we can do this all in-house. Therefore, I can get all of my data that I require quite easily with little fuss, and I can even just go and see the scientists and just tell them how are the results going. We also have great public engagement opportunities, so scientists can do outreach programs to schools and indeed the general public to really try and encourage individuals and enhance their understanding of what we do within the School of Life Sciences and the drug discovery unit, and really try and get, from a school perspective, young kids interested in science at a very early age. As for Vanaris and my industry sponsors, so we have an open dialogue regarding ongoing chemistry, which we can share the expertise and knowledge. But one of the major benefits of having an industry sponsor as part of your PhD is you get to carry out a placement. This is usually 3 or 6 months in length. As for me, this will be an invaluable opportunity to be part of a drug discovery project and team and gain invaluable experience, as well as a good skill set that will set me up quite nicely for future employment within the scientific research community. That's currently where I'm at the moment. So I'd like to acknowledge my supervisor, Professor Paul Wyatt, and Dr. Gary Tarver, as well as Bernardus Research Limited, the Medical Research Council, MRC, University of Dundee, and the Wellcome Centre for Anti-Infectious Research, and everyone for listening. Thank you.